Okay, so good evening everyone. Uh, Today is the 10th problem solving session for the course Experimental Biotechnology. The course instructor is Professor Vishal Trivedi from IIT Guwahati, and I am Shrija Ghosh, a PMRF from IIT Kanpur. So till now we have discussed about electrophoresis and then chromatography, and then we discussed about cell culture medium and everything. So now we are going to discuss a new topic, that is polymerase chain reaction. So we are going to see what is polymerase chain reaction, how it is done, what are the protocols and everything. Okay, so I'll be discussing certain questions regarding PCR, and while solving those questions I'll try to clarify as many doubts as possible on the topic. Feel free to ask anything whenever you have a doubt, like stop me at any point if you have a doubt, and I'll try to help you out as much as I can. Okay, so I'll start with the first question. Question number 1: what type of bond holds the complementary base pairs together in the DNA double helix? What type of bond holds the complementary base pairs together in DNA double helix? Option A, covalent bond. Option B, ionic bond. Option C, hydrogen bond. And option D, van der Waals bond. So what is the type of bond that is present between the base pairs of the two complementary bases in the DNA? Okay, we have one answer, option C, hydrogen bond. Okay, I'm waiting for the other person to answer as well. Okay, so both of you agree with option C, so that is hydrogen bond, that is the correct answer. So as you must be knowing that DNA generally is present in a double helical structure. It is a double stranded molecule. What do you mean by double strand? That means it has two strands that are complementary to each other. So one strand goes from 5 prime to 3 prime, and then the other strand is complementary, that is opposite to the 5 prime will be 3 prime and opposite to 3 prime will be the 5 prime strand. So basically the whole strand is the opposite of the whole strand is the complementary of that strand, and in this we have different base pairs. So we have 4 bases, A, T, G and C: adenine, thymine, guanidine and cytosine. Okay, and these have complementarity with each other. So A is complementary to T and G is complementary to C. Okay, so we have here you can see adenine and thymine are having complementarity to each other by making 2 hydrogen bonds. So this complementarity is because of the ability to form hydrogen bonds. So adenine can form hydrogen bonds with thymine and guanine can form hydrogen bond with cytosine. But if you see in adenine, like between adenine and thymine, the number of hydrogen bonds are 2, and the number of hydrogen bonds in guanine and between guanine and cytosine is 3. Okay, so it has 3 hydrogen bonds, it has 2 hydrogen bonds. What does it mean? That means G and C are paired more strongly than A and T. Okay, so by seeing the number of hydrogen bonds, so if we have a strand in which number of G and C's are higher than the number of A and T, that means it will be more strongly bound as compared to the strand that has more percentage of A and T and less percentage of G and C. Okay, as you can see, it has 2 hydrogen bonds and it has 3 hydrogen bonds. Fine. Moving on to the next question, question number 2. Which of the following is the correct order of stages in a cell cycle? Which of the following is the correct order of stages in a cell cycle? Option A, G1, S, G2 and M. Option B, G2, G1, S and M. Option C, G1, S, M and then G2. Option D, G1, M, G2, S. Which of the following is the correct order of the stages in a life cycle? Okay, we have one answer, option A. Okay, 2 people are agreeing to option A. Anyone else wants to answer? So option A is the correct answer. You guys are right. So in a cell cycle, basically we have G1 phase that goes to S phase, then we have the G2 phase and then we have the M phase. What are these phases? Basically when we have a cell, so let's say I have a cell, and I have the genetic material, that is DNA. In this case, the simplest example I am taking of a bacteria. I have a bacterial cell with the DNA inside. Okay. Now, if this bacteria has to divide into two bacterial cells with its own DNA inside it, what does it have to do? It has to go through a cell cycle. What does it mean? That means this bacteria will prepare itself to divide into two different cells. Okay, how is it going to do so? So basically, first it will prepare itself where it has, so basically one cell, if I have this cell, this is only going to divide into two cells, like it will expand in size, and after certain stage it will form two sets of DNA, and then finally this division is going to happen, which is going to give two different progeny cells or daughter cells. Okay, so one cell dividing into two cells. Now for that, first this cell has to expand in size to come into this stage, right? So first the cell is there and the cell has to expand in size. So for expansion, what will happen? The cell will increase its cytosolic space and everything. Okay, cell will kind of bulge in size. So that is what is going to happen in G1 phase. So this is a preparative phase. So this is a preparative phase where the cell prepares itself to enter into S phase. What is S phase? S phase is nothing but the synthesis phase. Okay, S phase is synthesis phase. What is what has to be synthesized here? We need two sets of DNA. Okay, so now the cell has expanded. So let's say the cell has expanded, but still it has only one copy of DNA. So it needs to have two different copies of DNA, right? So these two copies of DNA will be synthesized in the S phase. Okay, this is the synthesis phase. So basically what will happen? Replication of DNA will happen. Okay, replication of DNA will happen. So now once we have two copies of DNA, the cell will enter G2 phase, which is again a preparatory phase, where the cell will now prepare for the M phase. M is the mitotic phase. Okay, mitotic phase. So this was the synthetic phase, this is the mitotic phase, Mitotic phase means the cell will undergo mitosis. Mitosis means the cell will finally divide into two different daughter cells. Okay, one cell. So now the cell has expanded, it has two different genome. In the G2 phase, those two different genome will localize into two different cells, basically ends of the cells, and then in the mitosis phase the division will happen and we will get two different daughter cells with their own genetic material. Okay, so we have G1 phase which is a preparative phase for the synthesis phase. Then we enter the synthesis phase where the DNA replication occurs. Then the cell enters the G2 phase where again it prepares itself for the M phase, and in the M phase mitosis happens where the cell divides into two different daughter cells. Okay, so this is how the cycle goes: G1 to S to M, G1 to S to G2 to M. Okay, I hope that's clear to everyone. Okay, so I'll move to the next question now. Question number 3. In which phase of cell cycle DNA is duplicated? Option A G2, option B G1, option C M, and option D S. Right now we discuss cell cycle, so everyone should be able to answer this question. In which phase of cell cycle DNA is duplicated? G2, G1, M, S. Okay, again we have two answers, option D. Other people want to answer? I just now explained the whole cell cycle, so I hope you should be able to answer this. You can write the answers in the chat box. Okay, so option D is the correct answer. So as I explained, G1 we have in the cell cycle G1 phase, which is the preparative phase for the synthetic phase, that is the S. In the S phase we have the DNA replication where the DNA is duplicated and two copies of DNA are made. Then we have the G2 phase, and then finally we have the mitotic phase where the cell divides into two daughter cells. Okay, so option D is the correct answer. Moving on to the next question, question number 4. Dash prevents re-annealing of single strands of DNA during replication. Dash prevents the re-annealing of the single strands of DNA during replication. Option A helicase, option B single strand binding proteins, option C DNA polymerase, and option D primase. What prevents the re-annealing of single strands of DNA during replication? Okay, so Agatha says option B. Okay, so we have conflict between option B and option A. Anyone else wants to answer? Okay, so option B is the correct answer. Option A is helicase. So in DNA replication we have different steps. So as I told you, DNA is a double stranded molecule, right? So we have two molecules and DNA is present in a helical structure, right? So something like this. So first the cell, like when the replication is starting in the S phase of cell cycle, first the cell has to separate these two strands into two single strands. Okay, one will be 5 prime to 3 prime, the other one will be 3 prime to 5 prime. So cell first has to separate these two strands. So for that purpose helicase is used. So it basically separates the helix, okay, into single strands. Fine. After this, now these single strands can again re-anneal, because the attachment is basically hydrogen bonds, right? So again the and these two strands are complementary to each other. We have just separated it using an enzyme. So basically we have two strands like this, and we helicase enzyme is basically separating these two strands by breaking the hydrogen bonds. It is separating. Again these two strands have the capability of rejoining back, because they can again form the hydrogen bond. Nothing is stopping it to forming the nothing is stopping it from forming the hydrogen bonds. So to actually stop that, we have single strand binding proteins. Basically what these proteins do, these single strand binding proteins, they bind to these single strands of the DNA, and it actually prevents the hydrogen bonding from happening. So it prevents the hydrogen bonding. So it basically prevents re-annealing of the single strand. Re-annealing means they cannot again go and form the complementary base pairs. They cannot go and form the hydrogen bonds between each other. They cannot re-anneal. So it prevents the formation of hydrogen bond. Okay. Okay, so basically it prevents the re-annealing. Then comes the after the single strands. So now we had the double helical structure, and then the DNA is now separated into two different strands. Now we will have the next role, the next enzyme in action will be primase enzyme, that is here. What it will do, it will synthesize small RNA primers. Okay, small RNA primers that will bind to the starting region of the single strand. So it will bind here, and I made a mistake, just a second. So it will bind, it will make small RNA primers, and these primers will be complementary to this region. So basically this part and this part of the template strand, that is our genomic DNA, primase enzyme will synthesize primers complementary to these regions. Okay, and these strands, these complementary small primers will be in the 5 prime to 3 prime direction. So that's why now complementary strand for this part will be 5 prime to 3 prime, right? And this will also be 5 prime to 3 prime. Why I am emphasizing on 5 prime to 3 prime? Because the next player that is coming into action is DNA polymerase. What is DNA polymerase doing? DNA polymerase is basically going to extend these primers and synthesize the new DNA. It is going to synthesize the new DNA, and this synthesis can only occur in 5' to 3' direction. Okay, DNA polymerase can only synthesize DNA in 5' to 3' direction, and it can only synthesize DNA from an existing primer. That's why we need primers to be attached to the template strand, and then DNA polymerase will extend this primer and synthesize the whole new DNA. So once the whole replication is completed, we will have a one strand that is derived from the parent, the red one, and one strand that is newly synthesized, the blue one. Okay, so that is how in the next cycle of replication, again we will have one red strand and one blue strand, and then new strands will be synthesized against each of them. Okay, so do you understand the role of each enzyme? Helicase, it is separating the helix. Single strand binding proteins, it is preventing it from annealing back. DNA polymerase, it is the major enzyme that is going to do the polymerization, that is going to synthesize the new DNA, and primase that are going to synthesize the primers for this DNA. Okay. Yes, do you have a question? Saugata, you have raised your hand. Do you have a question? Okay. Okay, so the correct answer is single strand binding proteins that are preventing the re-annealing of single strands of DNA during replication. I hope the role of each enzyme is clear now. Anyone has any doubt how the replication process is going on? Okay, if it is clear, I think I can move on to the next question. Saugata, your hand is still raised. Can you put it down? I think it's clicked by mistake, probably. Moving on to the next question, question number 5. What is the purpose of the denaturation step in a PCR cycle? Option A, to anneal primers to the template DNA. Option B, to amplify the target DNA. Option C, to synthesize new DNA strands. And option D, to separate double-stranded DNA into single strands. What is the purpose of denaturation step in a PCR cycle? To anneal the primers to the template DNA, to amplify the target DNA, to synthesize new DNA strands, or to separate the double-stranded DNA into single strands. Okay, we have one answer, option D. Okay. So both of you are answering option D. Anyone else wants to answer? So option D is basically to separate the double-stranded DNA into single strands. That is the correct answer. So basically in the denaturation step, as I explained, PCR is nothing but a extended version of the replication. So whatever is happening inside our body, that process we have applied so that we can use it for different purposes, and we can do it in vitro, that is in a vial. outside the cell. So this is a process that is, the idea is basically taken from the replication process that is happening inside the cells. Fine. So in the replication process also, I told you there is an enzyme called helicase that is separating the double stranded DNA into two single strands. Now in PCR cycle, we don't want to put so many enzymes. It would become a very complex system with multiple enzymes. Okay. So what we do instead, we simply heat the DNA. So I told you the DNA are connected by hydrogen bonds, right? The two strands of DNA are having hydrogen bonds between them, between the two, between the different base pairs. Okay, so between adenine and thymine and between guanine and cytosine, we have hydrogen bonds present. So now hydrogen bonds can easily be like we can easily break the hydrogen bonds by heating it. Okay, these are weak bonds. Okay, so if we heat the DNA, what will happen? The hydrogen bonds will break and the DNA will denature into two single strands. So that is what we do in PCR cycle. The first step, like in replication also, we needed helicase to separate the DNA into two single strands. In PCR, what we do, instead of using an enzyme, we simply heat the DNA to separate into two DNA strands. Okay, and then we add the primers and add polymerase so that the rest of the replication process can go on and we will get an amplified DNA. Okay, so we will study about the details of PCR steps in the upcoming questions, but I hope this part is clear. So denaturation basically means separating the double stranded DNA into two single strands by breaking the hydrogen bonds between them. Okay. So option D is the correct answer, to separate double stranded DNA into single strands. I hope that's clear to everyone. Okay, I'll now move to the next question, question number 6. Dash of each DNTPs are added to the PCR reaction mixture. Option A 100 micromolar, option B 200 micromolar, option C 10 micromolar and option D 50 micromolar. Dash of each DNTPs, basically we have to tell the amount of each DNTPs that are added to the PCR reaction mixture. 100 micromolar, 200 micromolar, 10 micromolar or 50 micromolar. Okay. Option B 200 micromolar. Okay, we have one answer, option B. Anyone else wants to answer? So option B is the correct answer, 200 micromolar. Very good. So this is basically, I can say, like when you are doing the experiment, so these are some fundamental things that that has been established. So template DNA and primers, we will have to optimize exactly how much you need to do, you need to add. So whenever you are doing a PCR reaction, I told you, like any template that you give and you add the primers to it, so the polymerase can extend that primer to amplify the whole template. Okay, now if your template is very short, like if you have your template itself is let's say 4 or 5 kb long, that template you can add in lesser quantities as compared to large genomic DNA. So genomic DNA is a huge DNA molecule, right? So for such molecules, for larger DNA fragments or like genomic DNA, we need little higher amount of DNA as compared to the shorter templates. If we are amplifying something from a plasmid DNA, we can use it in lesser quantity as compared to the genomic DNA. Okay, so for plasmid DNA or for shorter templates, we can use DNA from 1 picogram to 1 nanogram. Okay, 1 nanogram of DNA is more than enough for amplifying. But if we are using genomic DNA, we need at least from 1 nanogram to 1 microgram. So we need at least like 100-200 nanograms in order to amplify a particular region from the genomic DNA. So generally, when we are using shorter templates, so let's say, just a second. So let's say we have a plasmid DNA, okay, a double stranded plasmid DNA, and I only want to amplify this region out of the plasmid DNA. So I'll design the primers for both the strands and I'll amplify this by doing PCR reaction. So for a plasmid DNA, I'll require lesser quantity, like 1 nanogram would be more than enough for that, or 0.5 nanogram would be enough. But if I have a huge genomic DNA, okay, and I only want to amplify a small region out of the whole genomic DNA, so for that I'll need larger amount of DNA, because this molecule is so big that the chances of getting denatured completely and the primers annealing to it reduces. Okay, so if I have just two small fragments like this, and if I have to separate, chances are higher for this to separate completely, right? But if I have longer molecules of DNA, what will happen? Maybe some part of DNA gets denatured and some part doesn't get denatured. Okay, so to ensure that all the strands are denatured, it's better to add it in higher quantity. Even if some strands are not getting denatured completely, or there is some problem with some strands, we will have it in excess, so that we can always rely that our amplification will happen, our reaction will be successful. Okay, so for larger DNA, we generally put it in larger quantities. Okay, then comes the primer. Again, the quantity of primer also has to be optimized. So generally 0.1 to 0.5 micromolar of each primer is enough. Generally we add 0.2 or 0.25 micromolar of primers, and it works for most of the PCR reactions. But if let's say if you added primer, like if you are doing a PCR reaction to amplify a particular gene from the genomic DNA, and you did not get the amplified product after doing the reaction, so you can check the amount of template or amount of primer. You can optimize that part so that you get a perfect amplification. Okay, then comes magnesium chloride. So why do we add magnesium chloride? So as you know, the polymerization reaction is going to happen by using a DNA polymerase. Okay, so generally for PCR reactions we use a thermostable DNA polymerase, that is a tagged DNA polymerase. So this tagged DNA polymerase, or any DNA polymerase in general, they need a cofactor of Mg2+. Okay, so DNA polymerase needs a cofactor, that is Mg2+ ion. So these are basically metalloenzymes. So Mg2+ ions are required by the enzyme. So we add additional magnesium chloride, because the enzyme that we get, if like we add magnesium chloride because the enzyme that we have doesn't have the magnesium ion present inside it, so we have to add it from outside so that it can use the magnesium ion, otherwise the enzyme will not be active. Okay, then we add the DNTPs. DNTPs are basically DATP, GTP, CTP and TTP. Okay, so mixture of all 4 nucleotides are added. Okay, so basically a mixture of all 4 nucleotides, that is DNTPs, is added, and that concentration is fairly fixed. 200 microliter of each DNTP is added. So 200 microliter of this, and this. Okay, and then finally we add the DNA polymerase, that is added like 0.5 to 2 units. Generally the lowest quantity is enough for the activity to occur, the like for the reaction to happen. Like 0.5 units is more than enough in a 50 microliter reaction. But sometimes if your reaction is not working, or if your enzyme is not efficient, like enzyme's efficiency has been lost, then you can add more amount of enzyme in order to get a perfect amplification. Okay, so this was about the amount of each reagent required for setting up a PCR reaction. Anyone has any doubt or any question in this? I hope that's clear to everyone. I'll move on to the next question. Question number 7. What is the purpose of final extension step in a PCR cycle? What is the purpose of the final extension step in a PCR cycle? Option A, to denature the DNA. Option B, to amplify the DNA. Option C, to add extra cycles to the reaction. And option D, to ensure all single-stranded DNA is fully extended. What is the purpose of final extension step in the PCR cycle? To denature the DNA, to amplify the DNA, to add extra cycles to the reaction, or to ensure all single-stranded DNA is fully extended. Okay, we have one answer, option B, to amplify the DNA. Okay, anyone else wants to answer? Come on, if you do, even if you don't know the answer, you can try to guess the answer. Like, think about it and answer something so that you will understand. Like, at least in this way you will be able to think about the question. So, anybody? So actually the correct answer is option D. So in a PCR cycle we have major steps, that is denaturation. As I explained earlier, what is denaturation? Then comes the annealing, where the primers anneal to the single stranded DNA. Then comes the extension, where the polymerase performs its function by extending the newly stranded DNA, basically extending the primer. So it keeps adding the different nucleotides using the different DNTPs, complementary to the single strand, ahead of the primers. Okay, and then after extension we have this next stage. Like, once this annealing and extension is happening, so this step will keep on repeating. So again extension, then again we'll come back to denaturation. So from the 2 strands that we have, now we will have 4 different strands, right? Now these two will go again separate, and then two new strands will form, and these two will again go and two new strands will form, and that's how the cycle will continue. So this will basically keep on going. So extension, then again denaturation, annealing, extension, denaturation, annealing, extension. So after the certain number of cycles, so let's say I wanted 30 cycles, so after 30 cycles we give an additional 10 minutes extension. Okay, we give additional 10 minutes extension, and this extension is to ensure that all the single stranded DNA is fully extended. What does it mean? That means, let's say like this cycle is going on very fast, like within 1 or 2 minutes we are denaturating the DNA, annealing the primers and extending it. Again we are denaturating the DNA, annealing the primer and extending it. Each cycle is hardly 1 to 2 minutes long. Okay, so each cycle is hardly 1 to 2 minutes long. So basically what does it mean? Let's say I had a single stranded DNA, primer is annealed, and now the polymerase is synthesizing the DNA in this direction, but before it could complete the synthesis, again denaturation started. So we have two strands, and again the primers will anneal, and then again extension will start. So if there is by chance there is some DNA single strand that has the primers annealed, but the extension did not get completed, in that case, in this additional 10-minute extension that we are giving, all those DNA extension can get completed that has not basically that did not get fulfilled during the cycle reaction that was going on. So additional 10 minutes we give for the polymerase to extend all the single stranded DNA that has not been fully extended. Sorry. Okay, so this is clear. So final extension step is to ensure all the single stranded DNA is fully extended. Is this clear to everyone? Okay, great. Then I'll move to the next question. Question number 8. Okay. Why is Taq polymerase particularly well suited for PCR? Option A, it is highly temperature stable and remains active at high temperatures. Option B, it is less expensive than other DNA polymerase. Option C, it produces longer DNA fragments than other polymerases. And option D, it is derived from eukaryotic cells. Why is Taq polymerase particularly well suited for PCR? So both of you have answered option A. It is highly temperature stable and remains active at high temperature. That is the correct answer. So I have already explained. So in the PCR reaction, I told you that we do the denaturation step, which is basically heating the DNA so that the hydrogen bond is broken. Now when we are heating the DNA, the reaction mixture has to reach up to 94-95 degrees Celsius. Okay, now if I want to do the reaction in a single vial without adding the polymerase again and again, and without adding different reagents again, I just want to add everything, put it in a vial, and then directly get the amplified product. I do not want to add everything in between after every 1 minute or 2 minutes. Then what will be the problem? Since all enzymes, most of the enzymes are heat labile, that is, they are not very they are sensitive to higher temperatures. So if they are sensitive to higher temperature, when we have a DNA strand that has to be separated, we have a double stranded DNA, and that double stranded DNA will be heated up to separate them into two single strands, right? Now in that heating step, when I'm putting the vial at 95 degrees Celsius, my enzyme is going to get denatured. It's a protein after all, right? So it will get denatured. So for that purpose, what people did was they got a unique polymerase from a thermophilic bacteria, that is a bacteria that can grow at higher temperatures. Okay, this bacteria is especially found in places that has higher temperature, and by higher temperature I mean really high temperature, temperatures above 50-60 degrees Celsius. Okay, so examples are hot water springs. Okay, so in such conditions there is a bacteria that is found, and in that bacteria, now that bacteria is surviving in hot temperatures, okay, higher temperatures, temperatures like 60-70 degrees Celsius. Now that means that that bacteria has enzymes that can function at those temperatures, right? A thermophilic bacteria that is growing at higher temperature will have enzymes that will be functional at those temperatures. So that was the idea that was used, and people isolated a polymerase from a thermophilic bacteria, that was Taq polymerase, and that Taq polymerase, it is isolated from the bacteria Thermus aquaticus, and that Taq polymerase is basically now stable at higher temperatures, and that can be added to the reaction vial at the start, and we can simply heat the reaction vial. Nothing is going to happen to that polymerase, and then that polymerase is going to perform its function again and again for all the number of cycles in a PCR reaction. Okay, in a polymerase chain reaction. So that is why we use Taq polymerase, because it is highly temperature stable and it remains active at higher temperatures. So we don't have to add the polymerase again and again when we are heating the DNA. Okay, the enzyme is not gonna get denatured. I hope that's clear to everyone. Moving on to the next question, question number 9. What is the primary advantage of using a hot start Taq polymerase in PCR? What is the primary advantage of using a hot start Taq polymerase in PCR? Option A, it reduces the annealing temperature. Option B, it enhances the specificity of amplification. Option C, it eliminates the need for primers. And option D, it speeds up the extension step. Why do we use hot start Taq polymerase in a PCR? It reduces the annealing temperature, it enhances the specificity of amplification, it eliminates the need for primers, and or it speeds up the extension step. What would be the answer? Okay, Arul Mozi has answered option B. It enhances the specificity of amplification. Okay, anyone else wants to answer? Okay, so option B is the correct answer. You are right. It enhances the specificity of amplification. So what is a hot start PCR? So basically, in a hot start PCR, what we do, we generate. So when we are doing a polymerase chain reaction, when we are setting up a PCR in a vial, so we have the PCR vial, PCR tube, in which we are adding all the reaction components, that is we are adding the primers, we are adding the buffer, and then what do we add? We add the template, and we are adding the DNTPs, and finally we add the polymerase, right? So all these things are added. So when all these things are added, they can actually start the reaction even if we do not put it in the thermocycler, put it like even if we don't start the reaction, we don't provide the temperature. At room temperature also, these primers can have some annealing efficiency, and the polymerase can function at the room temperature. So and what will happen, those reactions will not be very specific, because those reactions are not happening at its optimum temperature, right? So there will be non-specific reactions going on at room temperature, or even if the reaction vials are kept on ice. Okay, so to prevent those non-specific reactions, generally what people do, either they heat the thermocycler to 95 degrees Celsius and then directly put the vial in the thermocycler instead of keeping the vials in the thermocycler. So what we do is, we add everything and we take the vial and keep it in the thermocycler, we close it, and we start our program, where we when we start our program, what is happening, the thermocycler will gradually increase and reach the 95 degrees Celsius for the denaturation step. Now at that temperature, when it is reaching that temperature, it will pass through the 60-70 degrees Celsius temperature, and at that temperature polymerase can actually start its role before the denaturation has completed. Okay, so what will happen, because of those interfering steps, we will get a lot of non-specific reactions. Okay, so generally what we do, we generally keep the vial directly at 95 degrees Celsius, so that there will be very less non-specific reactions. So in the market we get hot start. So this is the hot start PCR, where we are starting at the higher temperature itself. We are not starting from the room temperature and then reach the 95 degrees Celsius. We directly start at 95 degrees Celsius. That is the hot start PCR, hot start polymerase chain reaction. Now in hot start Taq polymerase, what we get, so the Taq polymerase enzyme will have an inhibitor that will inhibit the enzyme to function at room temperature. It will only function at higher temperatures. So when the temperature will reach the 95 degrees Celsius, at that point the inhibitor will get denatured, and then the Taq polymerase will become active. So the Taq polymerase will remain inactive at room temperatures. So that is the hot start Taq polymerase. It will only start at a higher temperature. Okay, so this is how we enhance the specificity for amplification. We will not get any non-specific binding. So what do we mean by non-specific binding is, so let's say we have a genomic DNA. So genomic DNA will be very long strand of DNA, right? Now in this I only want to amplify this particular region. Okay, I want to amplify this particular region, but since these are DNA, so this primer can partially bind to some other region of DNA. Okay, and polymerase, if that is also active, it can start amplifying these regions also along with this region. So when we will analyze the amplified product on agarose gel, what we'll see along with this band. So you have studied about electrophoresis in the previous sessions, right? So in an agarose gel, when the sample is loaded in the well, we will see along with the band of interest, we will see a lot of non-specific bands, higher and lower in size depending on where the binding is happening, and we don't want that. We want only this region to be amplified in larger quantities so that we can use it for our further experiments. So for that purpose we have to reduce the non-specificity, we have to enhance the specificity, and for enhancing the specificity we use hot start Taq polymerase, which is nothing but a Taq polymerase attached to an inhibitor, which makes the Taq polymerase inactive at room temperatures and it is only active at higher temperatures. Is that clear? Both hot start PCR and hot start Taq polymerase. Okay, so we'll move to the next question now, question number 10. In PCR primer design, what is the significance of the GC content? Option A, high GC content makes primers strongly bind to the template. Option B, low GC content improves the primer stability. Option C, GC content has no impact on primer performance. And option D, GC content determines the primer length. What is the role of GC content in the PCR primer design? Option A, high GC content makes primers strongly bind to the template. Option B, low GC content improves primer stability. Option C, GC content has no impact on the primer performance. And option D, GC content determines the primer length. Okay, we have answers, two answers. Option A. That is, high GC content makes primers strongly bind to the template. Yes, that is the correct answer. So I have explained in the first question itself that we have 4 bases, right? Adenine, thymine, guanine and cytosine. So I have explained that adenine and thymine makes 2 hydrogen bonds, whereas guanine and cytosine makes 3 hydrogen bonds. Because of these 3 hydrogen bonds, like 1 hydrogen bond extra as compared to 80, higher GC content means we will have a stronger binding. Okay, so when 2 strands are bound to each other, if one thing is binding with 2 hands, so let's think about it like this. If I am holding something with 1 hand and if I am holding something with 2 hands, obviously things I am holding with 2 hands will have a stronger grip than holding it with 1 hand. Okay, so similarly when we have 3 hydrogen bonds, we will have a stronger grip as compared to 2 hydrogen bonds, and that stronger grip will make a stronger binding to the template. So when we are designing primers, so we have a long template and now I am designing a primer that will anneal to the starting part of the gene of interest wherever I am going to do the amplification. So this primer has to strongly bind to this region, otherwise what will happen if the binding is not strong enough, when the polymerase will, so first binding will happen and then the polymerase will come and do the extension step. So by the time polymerase is going to sit on the primer and then start the extension, if the binding is not strong enough, there is a chance that the primer can get dislodged from the template, the primer can fall off the template and we will not get the specific amplification of that particular gene. That's why we need a strong binding of the primer with the template DNA. Is that clear to everyone? Okay. So I hope that's clear. Moving on to the next question now. Question number 11. Calculate Tm for a 18 base pair primer with the sequence given. Okay, so we have a sequence from 5 prime to 3 prime here, and we have to calculate the Tm. What is Tm? Tm is the melting temperature. So what do we mean by melting temperature? So melting temperature, that is the Tm, it means that the temperature at which DNA strand, 50% of DNA strand will be in single stranded form. So if I have a double stranded DNA, the melting temperature will determine that 50% of this double stranded DNA is in single stranded form and 50% will be in double stranded form. So at the temperature, so I told you that when we increase the temperature, the primers or the double stranded DNA starts, so the double stranded DNA starts separating into single strands. So the temperature, when you are increasing the temperature, gradually this double strand will start separating into single strand, and there will be a particular temperature at which 50% of the strands will be in single stranded form and 50% of the DNA will be in double stranded form. Okay, so that is the melting temperature. So we have to calculate the melting temperature for a given sequence. So anybody knows how to do that, or anybody has calculated? I can give you 2-3 minutes to calculate if you know. Do you know how to calculate the melting temperature? Okay, so I'll give you the formula. So this is the formula basically. Okay, so for calculating the melting temperature, so I have this given sequence. So 4 degrees Celsius into the number of G and C's. Why higher Celsius for G and C? Because again GC bonds are stronger, so it will require a higher temperature to break the GC bond. So if I have a double strand, so here I'll have 3 hydrogen bonds connected, whereas here only 2 hydrogen bonds will be there. Okay, in the complementary strand only 2 hydrogen bonds will be there. So to break these we will need higher temperature, so 4 degrees Celsius for the number of G and C's in the primer and 2 degrees Celsius for the number of A and T's in the primer. So you can easily calculate. You just have to count the number of G and C's, multiply it by 4, and then count the number of A and T's, multiplied by 2, and then add it up. Okay, so now I am giving you 2-3 minutes to calculate. This is not very tough. Just count the number of G's and C's and count the number of A's and T's. Calculate this and tell me the answer, what you are getting. Okay, Saugata has calculated 50. Anyone else had calculated this? Anybody else wants to answer? So 50 is the correct answer actually. So we'll see how to calculate this. So we'll count the number of G's and C's first. So we have 1, 2, so I'll just put a dot here, 1, 2, 3, 4, 5, 6, 7. right? We have 7 G's and C's in the primer, so that means 7 into 4 degrees Celsius plus, now we have to see number of A's and T's. So we have, I'll put a cross here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11. So that means 11 into 2 degrees Celsius. So 7 into 4 degrees Celsius, that is 28, plus 11 into 2 degrees Celsius, that is 22. So what do we have here? We are getting 50 degrees Celsius. So the Tm for this strand, DNA strand will be 50 degrees Celsius. Okay, so you are correct. So here we see the option C is 50 degrees Celsius. So that is the correct answer. Okay, option C is the correct answer. So like this, if you have any strand, you can easily calculate the melting temperature by counting the number of G's and C's and counting the number of A's and T's, and then multiply the G and C with 4 and multiply the A and T with 2 and add it up. Okay. Moving on to the next question now, question number 12. Number of DNA duplexes obtained from one DNA duplex after 5 cycles of PCR are. How many DNA duplexes will be obtained from one DNA duplex after 5 cycles of PCR? Option A 4, option B 8, option C 16 and option D 32. Number of DNA duplexes obtained from one DNA duplex after 5 cycles of PCR. So duplex means double stranded DNA. So number of double stranded DNA we will get after 5 cycles of PCR if we start with one double stranded DNA. How do we calculate it? Anybody knows the answer? So I told you that in a PCR reaction, if we start with one strand of DNA, after one cycle it will be like one strand of DNA will separate into two single strands, one double stranded DNA will separate into two single strands and new strands will form against those. That means one double stranded DNA becomes 2. So after one cycle. So after 5 cycles, how many double stranded DNA will we have? Can you calculate it now? After one cycle we are getting 2 out from one double stranded DNA we are getting 2 double stranded DNA. So after 5 cycles, how many double stranded DNA we will get? Do we have 10 as options, Agata? So if I have a double stranded DNA, so what will happen? This double stranded DNA will separate into two single strands, right? And then new strand will form, right? New strand will form against these again. So this will be cycle number 1. So from one strand we have 2 strands. Again from each of these strands we will have 2 more strands, right? So in the second cycle what will happen? In the second cycle what will happen? We will get one green strand here, one green strand here, then we'll have the red strand here and red strand here, right? Again these two will separate into two single strands, and then these two will separate into two single strands. So second cycle will have 4 different strands, and then newly synthesized strands will be there. So that means after the second cycle we have 4 strands, right? Yes, you are right. After the second cycle we have 4 strands. So now you tell me after the third cycle how many strands we will have? Now you think after the third cycle each of these will separate into 2. Okay, so 2, 2, 2 and 2. So after the third cycle what is happening? So this is the second cycle. After the third cycle what will happen? How many strands are we going to get? Yes, we are going to get 8 strands. Very good. So now you understand how the progression is happening. So after 5 cycles how many are we going to get? After 5 cycles what would be the number of DNA duplex? (32). Yes, option D, 32 is the right answer. So basically what is happening, if we start with one strand, we are getting, after first cycle we are getting 2 strands. Then now after the second cycle we are getting 4 strands. So basically the progression is happening 2 raised to the power of n, where the n is the number of cycle. N is number of cycles. Okay, so after first cycle it will be 2 raised to the power of 1. So we have 2. After second cycle we have 2 raised to the power of 2, that is 4 strands. Then in the next cycle, the third cycle, we will have 2 raised to the power of 3, that is 8. Then fourth cycle we will have 2 raised to the power of 4, that is 16. Fifth cycle we will have 2 raised to the power of 5, that is 32. So every time after each cycle the number of DNA is getting doubled. That is why even if we start the PCR from one single strand of DNA, we can actually end up in a very high concentration of DNA after certain cycles. So you can see from one single strand, just after 5 cycles we have 32 strands. So if I start with 1 nanogram of DNA, then also I can reach higher amounts of, like I can achieve a lot of DNA, amplified DNA. That is why it is called amplification, because we are increasing the amount of a particular DNA fragment. Okay, so we are basically amplifying that to check the presence of that DNA, or to use it for further experiments. So if let's say I have 1 nanogram of DNA and I want like 10 micrograms of it, so I can simply amplify it using PCR and then use it for further experiments. So that is the purpose of PCR reaction. Okay, so because in PCR reaction we have the progression like this, 2 raised to the power n is the amount of DNA that we get. So if we start with one strand, we can get up to large number of strands after n number of cycles. So if we have n is equal to 10, that will mean we will get 2 raised to the power of 10 DNA duplexes. Okay, so that is how the amplification happens. Is this clear? So that is why the whatever the number of cycle is given, you just have to basically take the 2 raised to the power of number of cycles you have to calculate for that. Okay. I'll move to the next question now. So option D is the correct answer, 32. I'll move to the next question now, question number 13. When designing PCR primers, why is it important to avoid regions of repetitive sequences in the genome? When we are designing the PCR primers, why is it important to avoid the regions of repetitive sequence of the genome? Option A, repetitive sequences improve PCR specificity. Option B, repetitive sequences have higher melting temperature. Option C, primers may bind to multiple genomic location leading to non-specific amplification. And option D, repetitive sequences enhance primer template complementarity. What would be the correct answer for this? When designing PCR primers, why do we avoid regions of repetitive sequence in the genome? Anyone knows the answer? So Agatha says option D, and Arul Mozi says option C, primer may bind to multiple genes. Yes, option C is the correct answer. So just a second. Yeah. So if we have a long genomic DNA, okay, so and if we choose a region of repetitive sequence, so by repetitive sequence what do we mean? So if I have a whole genomic DNA, okay, and I want to amplify a particular region, I want to amplify this particular region, but the primer that I have designed, it is having the repetitive sequence in the genome. That means it can whatever the sequence I have chosen in this region, that same sequence is kind of present in other regions in that genome, okay, other locations in that genome. If that is the case, the primers along with annealing to this region, it will also anneal to other region, and if the primers are annealing to these regions, extension will happen, okay, genomic because we are just doing a polymerase chain reaction, so we are adding the polymerase, so wherever the primer is going to bind, polymerase will extend that region. So along with the region of interest, we will get different other amplification region, and again we will have non-specific amplification. So when we will analyze the DNA in gel, we will, along with the band interest gene of interest, we will get other bands on the agarose gel electrophoresis, which is because of the non-specific amplification. So that's why primer may bind to multiple genomic location if we use repetitive sequences in the genome for designing the primer. So that's why when we design the primers, we avoid the regions that have a repetitive sequence. We choose a unique sequence that is only complementary to the gene of interest or the region of interest. Like if I want to amplify this particular region, I'll choose the primer such that it is complementary to only that portion and not the rest of the genomic DNA, okay, to have minimal non-specific amplification. Is that clear? Good. So moving on to the next question. Question number 14. What is the main application of multiplex PCR in genetics and genomics research? Option A, DNA sequencing. Option B, gene knockout. Option C, simultaneous detection of multiple genetic markers. And option D, DNA purification. What is the main application of multiplex PCR in genetics and genomics research? Okay, we have one answer, option C. Anyone else wants to answer? Anybody else wants to answer? Okay. Again an option C. So option C is the correct answer, simultaneous detection of multiple genetic markers. So multiplex PCR is a technique where we can actually put multiple primers and amplify different regions simultaneously. Okay, so if I want to amplify multiple genetic markers or multiple genes at the same time, we can add the primers, all the primers for all the regions separately in the same vial. We can add all the separate primers, and in one reaction, like in one go only, all the genes will be amplified. So that is a multiplex PCR. Okay, so option C is the correct answer, simultaneous detection of multiple genetic markers. It is possible by using multiplex PCR. Moving on to the next question, question number 15. The length of the primers used for PCR should be typically between option A 18 to 24 base pairs, option B 500 to 1000 base pairs, option C 200 to 300 base pairs, and option D 1 kb to 10 kb. Length of the primers used for PCR should be typically between 18 to 24 base pairs, 500 to 1000 base pairs, 200 to 300 base pairs, 1 kb to 10 kb. Okay, both of you are answering option A, that is 18 to 24 base pair. That is the correct answer. So whenever we are designing a primer, first thing that we see is the length. So we do not want very long primers because it will take time for them to anneal properly. Also depends on our gene length also. So if we are interested in amplifying a DNA that is, let's say 1500 base pairs, and so for amplifying a 1500 base pair long template, if we take a primer of length between 18 to 24, that would be optimum. If we use a longer primer, let's say 500 to 1000, that means the primer is almost going to bind to the complete gene that we are trying to amplify. This binding will take a lot of time, so we cannot use higher size, any of these size range of primers. Second reason of not using longer primer is the cost, because primers we are going to get it synthetically made. So primer, we generally order the primers, and these primers are synthesized by using different DNTPs. Like these are short oligonucleotides that are chemically synthesized. Okay, so there are companies that can give you the primers. They can synthesize the primers. They have the synthesizers. So these are chemical synthesis. So basically they are making the chemical bonds by doing a chemical reaction. This is not a biological process. Okay, synthesizing a primer. So you just design the primer, you give them the sequence, they will send you the primer in a lyophilized powder form, and then later you can reconstitute it and use it for your reactions, use it for PCR. Okay, so using a optimum length of primer is best for getting a proper extension in the PCR, in the polymerase chain reaction. If you use very small primers, like smaller than 18 base pairs, let's say I have a primer of 10 base pairs, there's a higher chance that this primer will have specificity to other regions also. So if you increase the length to certain extent, it will be more specific, but if you increase it up to a very high number of base pairs, what will happen, it will again take time to anneal and it will be very cost inefficient. Okay. So the correct answer is option A, 18 to 24 base pair, as you have already answered. Okay. So moving on to the next question, question number 16. What should be the complementary DNA strand of the given strand? So we have a strand here 5' to 3'. You have to tell me what would be the complementary strand to this particular given strand. Okay, Arulmozi has answered option D. Swagata has also answered option D. Okay, let's see. So I have told you how do we write the complementary strand. So we'll write A against T and G against C. Okay, and the direction also we have to see. So basically if we have 5' here, the complementary strand will have 3', and then we start writing. So we have here T, so we will write an A, then A, so that will be T, then C will become G, G will become C, again opposite to G we will get a C, then A will have T, again T, G and A, and we will have 5' here. So you can match ATG, CC, TT, GA and 3' to 5'. So option D is the correct answer. So option D is the correct answer. That is, I hope, clear. Hello, am I audible? I think I lost my connection. Okay. My connection got lost. Just give me a second. Okay. So correct answer is option D. I hope this is fairly straightforward. So this is clear to everyone. Moving on to the next question. Again a similar question. What would be the complementary DNA strand from the DNA strand below? So again we have a DNA strand. So tell me the complementary DNA strand for the given strand. Okay. Option C. Both of you are saying option C. So let's see. now you have to again take care of the direction. Okay, so I'll write 5 prime, then I'll write T, A, G, 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 then after opposite to A we'll get T, then opposite to G we'll have C, C, then opposite to T we'll have A, then A will have T, C will have G, and then G, and then this will be 3 prime. So we have option C. Yes, option C is the correct answer here. So if you see in the option A and option C, option A and option C, the sequence is same, right? But you have to pay attention to the direction also. So when you are designing a primer, why I have added this question, so when you are designing a primer, you actually have to pay attention to the direction. So if I have a gene from 5 prime to 3 prime, if I have a strand, and now I have to design the primer, so I'll design the primer for the starting part. So when I am designing this primer will be 3 prime to 5 prime direction, right? Because this is 5 prime and wherever I'll end that will be 3 prime. So this has to be 5 prime and this side has to be 3 prime, right? I'll draw a longer strand for better clarity. So this is 5 prime and this is 3 prime, right? So if I am designing a primer of this region, this will be 3 prime to 5 prime. If I am designing a primer for this region, this will be 5 prime, sorry, this will be 3 prime to 5 prime here. Okay. So now for this region, when you are designing, when you are actually doing this experiment in your lab, when you have to order the primers, so you will only design the primers theoretically. Okay, after that you have to submit the sequence to a company that will give you the DNA synthesized, that will give you the primer synthesized. Okay, so to submit that sequence, you will have to send them the sequence in 5 prime to 3 prime direction. So when you have a gene sequence, you cannot design the primer for the single strand only. So I will have a gene sequence from 5 prime to 3 prime. The reverse primer that I have to design, so this strand cannot be complementary to this part. It has to be complementary to its reverse complement, right? Do you understand? So basically, just a second. So basically what will happen, I have a gene that is 5 prime to 3 prime, like I have a sequence, like this is a sequence, right? So the kind of similar sequence kind of something similar, I have a sequence like this. Okay, and now I have to design primers for this gene. Okay, so one primer I can directly pick the sequence from here and design a complementary sequence that will be 3 prime to 5 prime. The reverse primer has to be something that will attach to the complementary strand of this, right? So this is one strand, so this is the first strand and this is the second strand. The second strand will have 5 prime here, 3 prime here, right? So for this I'll have to design a primer complementary to this. So if I give you only this sequence, the first strand sequence, so what you will have to do, you will have to take this portion, first make the reverse complement of this portion and then design the primer in 5 prime to 3 prime direction. That's why you should know how to write the complementary DNA sequence and you should pay attention to the direction of the DNA sequence. Okay, so that is why these questions are important. I hope this is clear to everyone. I'll move to the next question now. What is the sequence for PCR process? uh, Swagata, you have again raised your hand. Do you have a question? Yeah, ask. By the previous question, answer is option D. Oh wait, I might have the answer is option C only. It's a typing mistake, sorry. I'll correct it when I'll upload the slide. Option C is the correct answer. Okay. Okay, I'll correct it when I'll upload the slide. Thank you for pointing it out. Moving to the next question now. Question number 18. What is the sequence for PCR process? Denaturation of the double-stranded DNA, DNA polymerase binds to the template primer hybrid, and annealing of primers with sequences that closely match with the template sequence, forming the primer template hybrids. So what is the correct sequence of the PCR process? Option A 1 2 3, option B 1 3 2, option C 2 1 3, and option D 2 3 and 1. So which step occurs first and which step occurs last? So we have three steps here: denaturation, DNA polymerase binding, and annealing. So both of you are saying option B, that is first denaturation is happening, then third process, that is annealing of primers, is happening, and then DNA polymerase binds. Yes, that is the correct answer. So option B is the correct answer. So as I have already explained, so we'll have a double-stranded DNA. So this is a double-stranded DNA. So first denaturation will happen, where we'll get two single-stranded DNA. So these will be single-stranded DNAs. Then in the next step, primers will anneal. So to this strand primer will anneal, to this strand also primer will anneal, and then in the last step extension will happen, where DNA polymerase will bind to the bound primer template hybrid, and then extension will occur for this strand in this direction and for this strand in this direction. Okay, so this is how the PCR reaction works. So this is denaturation, then this is the annealing, and this is extension. Okay. Okay, moving on to the next question now. Which of the following statements is true? Option A, PCR requires the target DNA, one primer, and all four deoxyribonucleotides and a thermostable DNA polymerase. Option B, PCR requires the target DNA, two primers, three deoxyribonucleotides, and a thermostable DNA polymerase. Option C, reaction requires the target DNA, two primers, one of the four deoxyribonucleotides, and a thermostable DNA polymerase. And option D, reaction requires the target DNA, two primers, all four deoxyribonucleotides, and a thermostable DNA polymerase. Which of the following statement is true? Okay, both of you have answered option D. Reaction requires the target DNA, two primers, all four deoxyribonucleotides, and a thermostable DNA polymerase. Yes, that is the correct answer. So I told you, you need the template DNA, that is the target DNA, whichever you want to amplify. You need 2 sets of primer, one forward primer binding to the top strand and one reverse primer binding to the bottom strand. Then you need all 4 deoxyribonucleotides, because you need, so you don't know what has to be incorporated, so you need all ATP, like DATP, GTP, DCTP and DDTP. Okay, so you need all 4 of them, and then finally you need a thermostable DNA polymerase like Taq polymerase. Okay, I hope that's clear. Moving on to the next question now. Question number 20. Taq polymerase is isolated from dash. Option A plant, option B fungus, option C bacteria and option D virus. This I have already told you. Taq polymerase is isolated from dash. Option A plant, option B fungus, option C bacteria and option D virus. Both of you have answered option C, that is bacteria. That is the correct answer. So Taq polymerase is basically isolated from a bacteria called Thermus aquaticus. Okay, so this is a thermophilic bacteria that is growing at higher temperatures. So it already has all the enzymes that are functional at higher temperature. Okay, so that's why we take the, so Taq basically comes from T of thermus and AQ of aquaticus. So we get the polymerase, the DNA polymerase of this organism is named as Taq polymerase. Okay, or Taq DNA polymerase. Okay, so option C is the correct answer. Moving on to the next question. Question number 21. PCR was invented by dash. Option A Carey Mullis, option B James Watson, option C John Hopkins and option D Hargobind Khurana. Who invented the PCR polymerase chain reaction? Option A Carey Mullis, option B James Watson, option C John Hopkins and option D Hargobind Khurana. Okay, both of you have answered Carey Mullis. That is the correct answer. So Carey Mullis was the one who had developed this technique, who had invented this technique, and this is a very useful technique in molecular biology and genetic genomics lab. Okay, so option A is the correct answer, Carey Mullis. Moving on to the next question, question number 22. Temperature cycles in a polymerase chain reactions are in the order. Option A 95 degrees Celsius, 60 degrees Celsius and 72 degrees Celsius. Option B 60 degrees Celsius, 72 degrees Celsius and 95 degrees Celsius. Option C 72 degrees Celsius, 60 degrees Celsius and then 95 degrees Celsius. Or option D 95 degrees Celsius, 72 degrees Celsius and 60 degrees Celsius. All of the temperatures are in degrees Celsius. Temperature cycles in the polymerase chain reactions are in the order of what? We were saying option A 95 degrees Celsius, 60 degrees Celsius and 72 degrees Celsius. That is the correct answer. So that so I've already told you about the like different steps in the PCR. So first stage we have the initiation, where we take our reaction mix to the 95 degrees Celsius time. So we give 5 minutes to reach the temperature up to 95 degrees Celsius and ensure that all the tubes are at 95 degrees Celsius. Then we have the denaturation step, where at 95 degrees Celsius all the strands will separate, the hydrogen bond will break, and from double stranded DNA we will get single stranded DNAs. And then comes the annealing steps, where we reduce the temperature from 95 degrees Celsius to temperature between 55 to 65 degrees Celsius. So that is the range for annealing, for primer to anneal to the DNA. So this annealing temperature depends on the Tm of the primer. Okay, Tm of the primer. So Tm of primer will depend on the length of the primer and the GC content of the primer. Okay, so if you remember the Tm calculation, the higher GC content will have a higher Tm and lower GC content will have a lower Tm, because we were multiplying 4 degrees Celsius to the number of G and Cs and 2 degrees Celsius to number of A and Ts. So if we have higher G and Cs, we will have a higher melting temperature. Okay, and as you know, we use 2 primers, right? We use 2 primers. So the Tm difference between 2 primers should not be more than 2 or 3 degrees Celsius. Okay, should not be more than 2 degrees Celsius. So Tm difference should be less than 2 degrees Celsius between the 2 primers, so that both of the primers can anneal to each strand at the same temperature. Okay, because we cannot have 2 different annealing steps, so we are doing the annealing step that is common for both the primers. So the melting temperature should be close to each other, so that the primers will anneal, and the primers generally anneal at a temperature that is 5 degree Celsius different from the melting temperature. Okay, sorry. Then we move to the elongation step or the extension step, where the polymerase, Taq polymerase, is going to sit on the primer template complex and extend that DNA. So this is the extension step, and then we keep on repeating this cycle. I told you, we again denature, anneal, extend, denature, anneal, extend. We keep on repeating this cycle till we get enough amount of amplified DNA. So this number of cycles can vary depending on your reaction, depending on your requirement, how much DNA you require. And then comes the next stage, where I have told you an extra extension time is given. So generally 10 minutes extension is given at 72 degrees Celsius for all the remaining single stranded DNA to get amplified, and then we reduce the temperature to 4 degrees Celsius for infinite time, so that the reaction will be stable. So whatever amplified product we get, so let's say if I am setting up the reaction at night, so I'll just set up this reaction. So you can see this step is 5 minutes, then this step is 30 seconds, 45 seconds and 2 minutes 30 seconds, and I'll repeat this cycle for 30, like I'll repeat this for 30 times. That means this will be approximately 3 minutes, this will be 3 minutes 45 seconds, right? So 3 minutes 45 seconds multiplied by 30, that means you can see this whole process will take around 1 to 2 hours, right? So I don't want to stay in the lab for 1 to 2 hours, so I can just set up my reaction and then leave it in the instrument. So when I leave it in the instrument, after 2 hours or after 3 hours, I can directly come and take out my vials. The vials will be at 4 degrees Celsius, so I don't even have to sit there, like as soon as the process ends, I have to take out my vials and keep it in the refrigerator for the DNA to be stable. I don't have to do that. I can easily program the instrument to reduce the temperature to 4 degrees Celsius, so that my products are stable. So this is called the final holding temperature, where we hold the amplified product. Okay, so this is the process of the whole PCR, whole polymerase chain reaction that can be performed in the lab. Moving on to the next question, question number 23. Why is PFU deemed more efficient than Taq polymerase? Why is PFU deemed more efficient than the Taq polymerase? Option A due to proofreading activity, option B due to superior polymerase activity, option C both A and B, and option D none of the above. Why do we prefer PFU polymerase more than the Taq polymerase? Okay. Arul Mozi has answered option C. Okay. And Swagata has answered option A. Option A is the correct answer due to proofreading activity. So, Taq polymerase has a major disadvantage. It doesn't have a proofreading capability. So, Taq polymerase actually lacks the 3' to 5' exonuclease proofreading activity. So, this is a process, like this is a property of the DNA polymerase that is present in most of the DNA polymerase. So, in humans also the DNA polymerase that is present in our body has this property, and this is there to ensure that whatever the extension is happening, that is accurate. So, if by chance there is some like instead of A, let's say guanine is added, instead of adenine guanine is added, so my body, so there will be a mutation in that case, right? So my body has developed processes where the polymerase itself can have a look at it, like if everything is okay or not. If it sees that okay, we have by mistakenly we have incorporated a wrong base, so we had to like we were supposed to incorporate adenine, but we have incorporated guanine. So what will the system do? It will cleave that guanine off and add an adenine to correct that mistake that it has made. So that is the proofreading capability of the DNA polymerase. But Taq polymerase unfortunately lacks this property. So it has a chance of misincorporating one base in 10 raised to the power 4. So a 400 base pair target will contain an error in 33% of the molecules after 20 cycles. So it's a very high error rate, and this error distribution is very random. As opposed to that, we have PFU DNA polymerase, which is obtained from another bacteria, Pyrococcus furiosus. So I've told you Taq is obtained from Thermus aquaticus, and we have PFU, which is another polymerase. It is obtained from Pyrococcus furiosus. So this DNA polymerase, this is also stable at high temperature, and this DNA polymerase possesses the 3' 5' exonuclease proofreading. So it is actually like a better polymerase to be used because the error rate is actually reduced in case of PFU DNA polymerase. We have the error rate reduced to 3.5%. So in Taq polymerase we had the error rate of 33%, whereas in PFU we have the error rate of only 3.5%. So it is going to give us more accurate amplified product. Okay, chances of having an error in the amplified product is lowered when we are using the PFU polymerase. Okay, this brings us to the last question of the session, question number 24. The primers used in the PCR process are dash. Option A, single stranded RNA nucleotide, option B, single stranded DNA oligonucleotide, option C, double stranded RNA oligonucleotide, or option D, single stranded this will be double stranded or DNA oligonucleotide. So we have two answers, option B or option D. Option D is now double stranded. I've changed the option. So you have changed your answer also. Yes, the correct answer is single stranded DNA oligonucleotide. So the primers that we are using are single stranded DNA oligonucleotides. So this brings us to the end of this session. So do you have any doubt regarding PCR? It's a very important technique. We use it to amplify any gene of interest, to either use it for further experiments, for cloning experiments, or to detect whether a particular gene is present or not, particular fragment of DNA is present or not. So if you remember, during the COVID times, we all were going through RT-PCR tests, right? So that detection was basically based on this technique. So we the viral genomic RNA, so COVID virus had RNA as its genetic material. Okay, so what we were doing, that RNA was first converted into DNA, and then they were using specific primers for the coronavirus. And if your sample has coronavirus in it, that primer will anneal to that sample, to that viral DNA, and you will see amplification. Okay, so that was the detection method for coronavirus COVID detection, and that is actually a very common detection method for most of the viral infections. It is very easy to design the primers for a viral DNA. We will design the primers for the viral DNA. The primers will go and anneal to the DNA, viral DNA, or complementary viral, like basically the complementary DNA to the viral RNA, and that primer, once it anneals to the cDNA, we will see the extension happening. And if you see an amplified product, that means your sample has that particular virus. If you don't see an amplified product, that means your sample doesn't have and that particular virus. So this is a test that is done for HIV also. This is a test that was being continuously used during COVID times. So all of you must have heard about RT-PCR. So today you learnt about the technique. I hope everything is clear, how the technique is done, what are the different steps, how the technique was developed. Like the idea came from the basic DNA replication that is happening in our body. So anybody has any doubt? Everything is clear? Okay. So if we don't have any doubt, I can end this session now. Thank you all for joining. I'll see you in the next class. Thank you. Bye bye.